AMALearning, you're about to. Specification point 1.13b. Core practical. Investigate the use of chemical reagents to identify starch, reducing sugars, proteins and fats. To successfully answer any GCSE question linked to this specification point, you will need to know the names of the chemical reagents and processes through which they help to identify starch, reducing sugars, proteins and fats within food. As with every core practical, it is important that we understand the process in detail. As this is a triple-only practical, it can be assumed it is more likely to come up in the exam. But nonetheless, the process must be fully understood, as would be expected with any core practical. Before we can test a substance, we need it to be in a solution first. By this we mean the solute, which in this case is starch, reducing sugars, proteins or fats, has dissolved into a solvent, a liquid such as water. Substances such as milk and juice are already in solution, and so can be tested as they are. Substances such as bread are not in solution, and therefore before we can test them, we must grind them into a paste with the pestle and mortar, and then add them to water in a beaker and stir. Questions relating to the test for starch is common, and hence knowing the process and the outcomes is important. Iodine is an indicator that turns bluish-black in the presence of starch. It is harmful, which means when using it. It is important to wear your safety goggles and avoid contact with your skin. When testing for starch, add iodine into a food sample. If the food sample doesn't contain starch, the sample won't change color. If the sample does contain starch, it will turn into a bluish-black color. Questions relating to the test for glucose is common, and hence knowing the process and the outcomes is important. When testing for glucose, add Benedict's reagent to a food sample. After heating it, if it does not contain glucose, the sample will not change color. If after heating it, the sample turns red, brown, orange, yellow or green, it means it contains glucose. The darker the color or the redder the color, the more glucose it contains. Questions relating to the test for protein is common, and hence knowing the process and the outcomes is important. Biuret reagent is an indicator that turns purple in the presence of protein. It is harmful, and therefore it is important to wear your safety goggles when using it, and also to avoid contact with your skin. When testing for protein, add Biuret reagent to the food sample. If the sample does not contain protein, the color will not change. If the food sample does contain protein, then it will turn purple. Questions relating to the test for lipids is common, and hence knowing the process and the outcomes is important. When testing for lipids, add ethanol and distilled water to the food sample and shake. If the food sample does not contain lipids, then the color will not change. If the food sample does contain lipids, it will turn a cloudy white. In addition to the method just mentioned, you can also rub the food sample on a piece of paper. If it does contain lipids or fat, it will leave a grease mark. Our animations were designed by experienced UK-trained teachers. We have been achieving amongst the best results in the country for many years, and we decided it was time to share our success. Have you just AMALed and want to AMAL some more? Don't forget to like, subscribe and share to keep yourself up to date with the latest uploads. AMALearning, you just did.